Good evening, everyone. I'm Katie Doyle Myers, the Executive Director of the Desmoid Tumor Research Foundation. I'm so pleased to be with you this evening for our webinar about navigating treatment options for desmoid tumors and updates on one of those, Zolucatetide, also known as the drug FOG-001. We are really excited for this conversation with Dr. Gregory Cote. Before I get to a formal introduction of Dr. Cote, let me tell you a little bit about where we're heading this evening. As many of you know, one of DTRF's core priorities is to provide patients, caregivers, and clinicians with access to current, consistent, and evidence-based information and education to help support informed decision-making and high-quality care. And that's why we're here this evening. So our objectives for this evening are number one, to walk through the current landscape of treatment options for desmoid tumors. We are also going to speak about the role of clinical trials in addressing our disease. We're going to look at key therapies, including GSIs and TKIs, and how each one of these fits into the clinical care today. And Dr. Cote will talk more about what those are. And then finally, we'll share emerging data on another targeted therapy, Zolucatetide, again, also known as FOG-001. We're going to use both of those names throughout this webinar. We'll talk about how the therapy works and what the early clinical findings are beginning to show. As a reminder, Dr. Cote can speak to the science and publicly available information, and there are certain sponsor-specific questions that he's not able to address. They would need to be addressed directly by the trial sponsor. In this case, is Parabolis Medicines for the Zolucatetide medicine. We'll keep reminding you of that, and we'll do our very best to answer all your questions. Throughout the discussion, our goal is to support more informed, shared decision-making, to help patients and care teams think through the potential risks, benefits, and trade-offs when considering different treatment options. So we have people coming in. We're so glad you all are here. We're going to get started with Dr. Cote in just a moment. I just wanted to say we are so grateful for this community and all of the questions that we've already received from registrants for this webinar. Please know that we reviewed each and every question, and we're going to do our very best to address as many as possible today. Unfortunately, we cannot address every specific case as much as we would like to. If you do not have your questions answered today, you're welcome to reach out to us at info@dtrf.org, and we will respond with appropriate resources. So also, if you're just coming in, welcome. I'll just review a little bit about how our time will go this evening. So the Q&A is open. Please write your questions for Dr. Cote there. After his presentation, we'll have time for a question and answer session. We will be monitoring the Q&A and we'll do our very best, like I said, to address as many questions as possible. Let's see here. Also, the chat function will be on for you to communicate with one another, to network, but this will not be monitored for questions during this session. You're welcome, again, to connect with one another and to network, but the chat will not be monitored. A really important announcement related to the chat, especially since we are talking about a current clinical trial. If you are participating in a clinical trial at this time, including the FOG-001 Zolucatetide trial that we'll talk about specifically, please refrain from disclosing that information in the chat to protect your privacy and the integrity of the trial. All right, we're almost there. So also, if you know someone who wanted to join but weren't able to be here this evening, this recording will be on our website, on our video library very soon by the end of this week, and we will notify the community, including all of the registrants for this webinar when it's public. You can also always subscribe to our newsletter at dtrf.org for the notifications. All right. So at this time, I'm so pleased to finally introduce you to our presenter this evening, Dr. Gregory Cote. Dr. Cote is a DTRF Medical Advisory Board member and a medical oncologist at Massachusetts General Hospital Cancer Center, in the Henry and Belinda Termeer Center for Targeted Therapies, the Center for Sarcoma and Connective Tissue Oncology, and the Stephan L. Harris Center for Chordoma. I apologize if I didn't pronounce some of those names correctly. I need to do some more homework on those. Dr. Cote's primary research interest is in developing novel agents for connective tissue malignancies through translational studies and early phase clinical trials. Dr. Cote, thank you so much for taking the time to join us this evening, for preparing this presentation, and for being willing to take questions from our community. Passing on to you now for your presentation. Go ahead
All
and
right.
share your screen.
All right, great. Thank you for the introduction. Let me get my slides set up here.
Sure thing. Take your time.
Let's see if we get this right. Took us a few times on practice. So how does that look?
I believe in you.
Can you guys
It's
see that okay?
looking great.
All right, great. All right. Well, really nice to be back again. Really happy to be here. And for those of you who caught the webinar last year, we're going to kind of refresh some of that and kind of build on some of what we talked about. At that time, we were really just introducing this concept of what FOG-001 was. And so now I can show you some of the clinical data, which hopefully you'll find pretty exciting. All right, let's get these slides to work. All right, so just a quick outline. You heard a little bit about what we're gonna cover. We'll go over desmoid tumors in general, some of the biology behind them and challenges in developing therapies for desmoid tumors. I'll take you through some some treatment options, including clinical trials. And then we'll get into the Zolucatetide trial, or FOG-001. And then at the end, I showed this last year, but I think it's really nice. It's sort of a roadmap of how I kind of think about a desmoid patient when I'm meeting them in clinic for the first time and when we're working through the different treatment options. And then we'll have plenty of time for questions at the end, as you heard. All right, so what is desmoid tumor? So I think this group probably knows this well, but this is a tumor, it's not a cancer. So this is a little bit of semantics, like what does this really mean? I think of like a cancer as like a malignancy, something that's high grade, that can travel to other parts of the body and cause trouble. Desmoid tumors, they don't do that, but they can grow and they can cause trouble in the area that they're in. And this could be pain, could be interfering with function of an organ, kind of crowding things out. But they don't travel and they're not really truly a cancer. So it's a tumor, quote unquote. We think these arise from these cells called fibroblasts. Fibroblasts are scar-forming cells. And if you think about, you've probably heard of some stories of patients who undergo a surgery and then develop a desmoid tumor afterwards. So anytime there's an injury to the body, there's a huge proliferation of these cells. And as they replicate, if one of them undergoes a mutation in this pathway called the Wnt beta-catenin pathway, and I'll show you that a little bit later, then they can become a desmoid tumor. Now, when we're thinking about therapies, it's really important for these next two bullet points. These are rare tumors, relatively speaking. It's about 5 cases per million people per year. And then tied to that, they have a what we call a heterogeneous clinical behavior, where some desmoid tumors can kind of grow and just be really slowly progressive over time and not really cause much of an issue. Some can grow and spontaneously regress. We saw that in the clinical trials in patients who got placebo and their tumors still regress. That's rare, but it happens. And then there's others where they can grow and grow and grow and grow and cause problems. And because of that, it's a real challenge when we're trying to develop clinical trials, because you have a smaller population to look at, but even within that population, the behavior can be just different. When we think about other challenges, this is rare diseases in general are a lower priority for big pharma. Big pharma, they have a lot of investment. They're looking for more common cancers, things that have tens of thousands of patients, not just thousands, and that can be hard for us to get an investment sometimes in this disease. And then the last challenge I'll say here tied to this is that this Wnt beta-catenin pathway that I mentioned earlier is a very difficult target to go after. There's these very specific kind of protein-protein interactions that aren't classic targets for drugs. So that's been a challenge. Now, that being said, we found some great partners in biotech and small pharma, and that's led to the successes that we've seen in the gamma secretase inhibitor space with nirogacestat and varegacestat coming down the pipeline. And I think we've really kind of proven to the industry side, investor community, that we can develop drugs in rare diseases. So we're making progress. And then tied to this, it does take a little bit of luck sometimes. So I'll just digress here for a moment. In all the rare cancers that I take care of, sometimes we have some overlap with more common cancers. And it turns out that the Wnt beta-catenin pathway is really important for colon cancer, one of the most common cancers. And then another common cancer from the liver called hepatocellular cancer. So these are much more common diseases, thousands of patients. And so if a drug company is looking to develop targets for this Wnt beta-catenin pathway for those other diseases, we can get sometimes involved in that too. And in the case of Zolucatetide, when Parabolis, the company, first came to Mass General to talk about their trial, this new drug they had for colon cancer, as part of my role in the phase 1 group, I just happened to be at one of these meetings where they were there, and they started talking about beta-catenin and colon cancer, and I raised my hand and I was like, "Have you heard about this disease called desmoid?" And like, "No, what's desmoid?" And we kind of got the ball rolling. I think we would have got there in the end because there's a lot of smart people in this space, and they would have thought of this idea too. But it allowed us to really get involved in this trial from the beginning. And in fact, the first patient who had a response where the tumor shrank was one of our desmoid patients. So it's a pretty neat story, and we just in some ways it was just a little bit of luck. So anyway, enough of that. All right, so we're going to take a little detour here. I showed these slides last year, but I think it's important to kind of go over it just to set the stage. And we're going to take you back to biology class here. And these terrible cartoons are my design of a cell in your body. Okay, so cells have a membrane that's depicted by this double line here, and there's sort of an inside of the cell and an outside of the cell. And inside the cell, there's a lot of machinery here that I'm not showing you. And very importantly, in the middle, there's this thing called the nucleus where your DNA is. And that's the programming that makes you you. And in fact, every cell in your body has the same DNA, but every cell might look a little different. And why is that? Well, there's different genes that are turned on and off in the course of that cell's life that makes it look like a skin cell, makes it look like a muscle cell, things like that. So in an advanced organism, it turns out cells need to talk to each other. So how do they do this? So there's a few different ways to do this. The most common way is something called a ligand and a receptor. And you can think of this as sort of a lock and key mechanism where some other cell is sending this ligand signal that only fits this receptor, which is the lock here. And once that happens, it starts a cascade of signaling within that cell. It's sort of like a game of telephone where one goes to the next, to the next, to the next. And then that turns on some program within the cell to make it do something. And in this case, I just wrote cell growth, but it could be something else. So there's these sort of gas pedals and brakes to these systems. So gas pedals are like the ligand telling it to happen, but there's also brakes that say, hey, we've had enough of this signal, we got to stop. We don't want to grow too much. And so there's these, we call them inhibitory pathways. And I just kind of oversimplify this, but there's a lot of complex mechanisms to do that. And so these are in a constant balance and they're very critically regulated. And it's not hard for you to imagine that in cancers or tumors, something could go wrong really at any step of the way here. And that could lead to a cell growing when it shouldn't grow. So you could have too much ligand, you could have a problem with the receptor, you could have a mutation along the pathway, or you could even have a mutation in the brake pedal system. And so in this example, I just put a mutation along that signaling cascade, and now it's stuck in the on position. So that gas pedal is pressed even though it shouldn't be. There's no ligand here, and that cell is being told to grow, grow, and grow, and grow. So if you ever heard the term targeted therapy, it's a common term in drug development. We're really trying to find targets to block these pathways sort of downstream, we call it, downstream from the mutation to stop it from happening. All right, and this is an example of a mutation in a brake pedal, so we'll skip that. All right, so let's pivot now to the Wnt beta-catenin pathway. So this is the pathway that I mentioned earlier. It's critical for desmoid tumors and other cancers. And the ligand here is called Wnt. The receptor's got a funny name called frizzled. And the downstream effector here is beta-catenin. So in the off setting, so Wnt is not bound to the receptor, beta-catenin is targeted by that brake pedal system, where APC is important. We'll come back to that later. That constantly recycles beta-catenin to the recycling machinery within the cells and says, so no signal, beta-catenin is made and it's degraded, made and degraded. When Wnt binds, APC falls off through a little tag that happens there, and beta-catenin goes to the nucleus where it turns the right genes on for that cell to function. So off, it's degraded, on, it's present. So in desmoid tumor, one of two things can happen. You can have a mutation in beta-catenin, so that even in the absence of Wnt, APC and its recycling machinery can't bind to beta-catenin, so you get too much of it, so it accumulates over time, and that leads to things getting stuck on where they shouldn't be on. Or the other way is to have a mutation in APC that even though beta-catenin is there and it should be degraded, it's not getting degraded. So there's two pathways to drive this. So if we pivot now to thinking about our therapies that we have, and this is in no particular order, none of these really target this pathway. They're sort of all indirect ways to hit the desmoid cells. So we have chemotherapy, so we have different drugs whose job it is to interfere with cell function. Most of these target DNA, where they try to disrupt DNA. And the idea there is you're trying to have what we call a therapeutic window, where you kill more of the desmoid cells, but not enough of good cells, so you can tolerate the therapy and the desmoid gets treated. We have some that are what we call kinase inhibitors. And sorafenib may sound familiar to some of you, because this is we had a positive study that showed that this works in desmoid tumor. These work a little differently. They're not really hitting this pathway. It's more after these genes are turned on, there's different programs that are active that are involved in how those cells grow, and the kinase inhibitors kind of block those programs. So we call it sort of downstream from the signal. We have these gamma secretase inhibitors, which gamma secretase is part of another parallel pathway here that's very important for desmoid tumors. We know this because the drugs work quite well. But it's not directly involved in beta-catenin. There's some like what we call a crosstalk between those pathways, but it's more of an indirect way to target the tumors. And then we have things like radiation and ablation. So radiation tries to disrupt these cells through something called ionizing radiation, where it tries to kill the cells. And then ablation does this with typically like a thermal treatment. So it's either really hot or really cold to try to break this cell membrane and kill the cells. So all these are quite different, but none of them really gets at the heart of the biology here. And a dream for all of us who've been treating this disease for a long time is if we could find a drug that could safely block this beta-catenin pathway, then we could find a drug for desmoid tumors. Okay, so then what is FOG-001 Zolucatetide? So a little different picture here. So this is just a zoomed in picture of a piece of DNA. DNA is a double helix, you might remember. And in the nucleus, we have lots of beta-catenin, and it binds to another protein called TCF, and that turns genes on. This is a biology symbol for a gene being turned on. It's just a little up arrow. And so what zolucatetide is a peptide, so it's a protein that's been engineered to be very stable. There's something called a staple here. So basically it's a way with this peptide to hold its shape that makes it very stable in biology where it otherwise would fall apart. And zolucatetide actually binds beta-catenin and blocks this interaction with TCF, so that even though there's a ton of beta-catenin around here, it can't bind to DNA and turn those genes on. This is a 3D model that Parabolis provided us that has beta-catenin, a really large protein here, and then zolucatetide, you can see, is a very, very tiny little peptide that just kind of shoves itself in the right spot. So really amazing technology here. I mean, you know, if you'd asked me 5-10 years ago if this was possible, I'd be like, oh, that's interesting, but I don't know. And yet we've seen here that this biology has been shown that they can do it. And I think it's really exciting now too, if you think about even diseases other than desmoid tumor, because we have a lot of, you know, very hard to treat sarcomas that have similar kind of protein targets that are hard to target with drugs. And now that we can see this here, maybe the same technology can be extrapolated to other diseases. So it's kind of an exciting time. All right, so we're going to take a detour here. We're going to talk about clinical trials, and I'll come back to the zolucatetide trial in a moment. I spent a lot of time on this last year, but I'm going to kind of pare it down now just for the sake of time. But if you're interested, there's another video on this where I go into a little more detail about what phase 1 trials are. But in general, when we're thinking about an oncology clinical trial, this is typically for cancer, but also for tumors. We're trying to match a drug with a patient who has a problem, has a tumor or a cancer. And this can take different forms. It could be a brand new drug, first time it's ever been in people. And this is called a first in human study, or phase 1. And nobody's ever gotten it before. And we're trying to figure out, is it safe? Does it work? I'll show you more of that in a second. Or it could be an existing drug that maybe is approved for a different kind of cancer. And we want to know, does it work in other tumors? So that's sort of a different path. And trials can be sponsored, we call them, by companies, could be by oncologists, academic centers, could be the National Cancer Institute. So there's different ways to kind of initiate these studies. Okay. So this is sort of the high-level view of the journey of a drug from preclinical research to FDA approval. So this is quite a process, and I'll take you through it at a high level. But things start in the lab where we're trying to say, okay, we have a new compound, it hits a particular target, and we think this is really important. So, maybe we're treating a certain kind of cancer and we want to block a certain pathway. We hit that, it looks good. Now we want to bring it to people. So this drug may look great in a mouse, and we may know the dose in a mouse, but people are not mice, right? So we got to find the right dose that is safe. It's the right dosing schedule, and we want to make sure it works, and we want to know the side effects. So that enters into what's called phase 1 clinical trials, or first in human. Typically, these are very small studies, dozens of patients, whereas like a phase 3 could be hundreds of patients. Usually they will include all different kinds of cancers, but there's some nuance there. So if you had maybe a new chemotherapy drug and you weren't sure what kind of cancer it might work in, and there's no specific target, any kind of tumor with any kind of mutation could really participate. But if you had a very specific drug that you really wanted to look at only in tumors that have this mutation, so if we take beta-catenin, pathway altered tumors, we only want to include tumors maybe that have that mutation, but it could be all different types: colon cancer, liver cancer, desmoid, other things. And so there could be some restrictions. So we start these studies with just a few patients at a very low dose, and then we'll slowly increase that over time. And eventually, as we get more experience with the drug, we know the side effects, we know the dosing, and hopefully we see signs that it works. As this drug matures during phase 1, we enter in something called phase 2. And these are larger studies, and typically it's a more homogeneous population. We have more of the same type of tumor, more patients with the same mutation. Typically, they're getting the same dose. And we're trying to build this story. Does the drug work? Are the side effects the same as they are in phase 1? Are we seeing what we want to see? And we're really trying to use this early part in time to make sort of this go, no-go decision before we invest a lot of resources in a big phase 3 study, because we don't want to waste your time. We don't want to waste, funding that could be used for other drug development if a drug's not looking like it's going to work. So if things go well, then we go to the later phase studies. And very importantly, so in phase 1, phase 2, there's typically not a placebo. Everybody gets the drug. There are some nuances there with phase 2 studies where there can be some placebos. But for the most part, phase 1 and phase 2 studies, everybody's getting the drug. Phase 3 is different. These are the ones that you hear about in the news. They're very large studies where you're comparing the drug versus either some prior therapy, some standard therapy, or a placebo. So this is called a control arm. Often these are blinded, like we don't know, what you're getting, the doctor and the patient, and that's what's sometimes required to really prove that a drug works. If it does work, then it can lead to FDA approval, and then we have a new agent to treat. This takes a lot of time, a lot of resources. We all want to accelerate this process. We're all trying to move it forward, but as fast as we can. But there's just the reality that it takes some time. All right. So key points here before we move on. For trials, this is an informed consent process. This is your choice to participate with your oncologist. You're not a guinea pig. If you look through this and you say, "Oh, that looks interesting, but it's not for me. Thanks, but no thanks." That's okay. You can tell your doctor that. They're still going to be your oncologist. They're not going to not treat you. Okay. So you need to be a participant in this. These early phase studies tend to be quite busy. They have a lot of visits. We have to do extra safety visits. We have to do extra blood draws to measure the level of the drug in you. Sometimes we have to do biopsies, things like that. The trade-off, though, is you get access to cutting-edge technology that otherwise maybe you wouldn't have access to, and you're going to get that drug. It's not placebo. Later phase studies, they tend to be less busy. We have more experience with the agent. We know the side effects, so the schedule's less intense. But there is that possibility that you could get randomized to the experimental versus the control arm, and we don't have control of that, and it could be a placebo. So that's obviously a really important consideration to think about. All right, and then as I mentioned earlier, it's often blinded. So okay, all right. So I think we're doing okay in time. So let's talk about the Zolucatetide study. So this is a phase 1, phase 2 study. So the first part of the study was when we were doing that dose escalation time, trying to find the right dose, all different kinds of patients. And then phase 2 is as we've gotten to understand this drug more, we're getting more experience with this drug, trying to optimize dosing, trying to see the effect that we want to see. Okay, no placebo in this study. All right. And as I mentioned to you earlier, this is part of a bigger study with different kinds of tumors, right? This Wnt beta-catenin pathway is really important for other cancers. And so here's listed as FOG-001, but it's the same drug, Zolucatetide. And part 1A here was when we were escalating the drug, trying to find the right dose for the drug. And included in this, there's some abbreviations here, but we're colon cancer patients and other patients with this Wnt beta-catenin alterations. And these included desmoid tumors, but if there was like a liver cancer patient who had this or some other rare cancer, they could also participate in this part of the study. So here is the drug by itself, trying to find the right dose. And then as we learned more about desmoid and the possibility that this drug could work in this space, we opened up a separate arm that was for desmoid patients only with what's called dose optimization. And this is more of that sort of phase two kind of structure where patients were in an arm specifically for desmoid tumors, so only desmoid patients, and we were giving different doses to try to optimize the dose, to try to fine tune it, because the dose for a patient with desmoid may be different than it might be for another type of cancer. Other arms that we're not going to cover today, but just so you know that they were open. So there were arms for colon cancer and other cancers, and then there's some other combination arms for looking at this drug combined with other chemotherapy agents that you might use for colon cancer as well. But we're going to focus on this. So the patients, the data I'm going to show you are going to be just for these patients that we had a cutoff of 2025. So you might be saying to yourself, oh, it's 2026. I want to see updated data. But these things take time, and it takes time for the data to be cleaned and correctly analyzed. So there's always a bit of lag time before between stuff we can present and stuff we have available. So just stay tuned when we want to share data as soon as it's ready. All right, so on to the trial. So here's some data, and you're probably looking at this bar and wondering what I'm showing you. So these numbers and these rows are individual patients, and you'll see on the bottom here these are weeks on treatment. So each number represents a person who participated in this trial. The colors I'll show you in a second are different doses, and this is the time that they were on the drug. So if we think about patient one, that's that first patient I mentioned to you who started at the second dose level during dose escalation, started at time 0, and as of this data cutoff at over 90 weeks was still on the treatment. The arrows mean that they're still on. And patient 2 started at time 0 and also still on almost 50 weeks. Patient 3 over 40 weeks, and so on. What are the circles and squares? So we have this way of measuring tumors in solid tumors called RECIST. You can see it vertically written here. And this is not a great way to measure tumors, but it's desmoid tumors, but it's the accepted way from the FDA. And basically, we take a caliper and we just measure a cross-dimensional size of the tumor, and we see how that tumor changes over time. And on this plot on the right, it's called a waterfall plot. Again, these are the same individual patients. So patient 1, patient 1, patient 2, patient 2. If this bar goes up, then the tumor grew. The bar goes down, then it shrank. And I think your eye will probably be drawn immediately to seeing that all the patients had tumor shrinkage, which is really nice to see with an early clinical trial. Yeah, I do a lot of phase 1 early phase studies. Been doing this for a long time, and I can't think of another study, maybe outside of one other one, where we saw something like this. So pretty remarkable to see that. And these circles and squares have to do with something called a partial response. See this PR thing here? And by RECIST, if it shrinks more than 30%, that's what this dotted line is, it's called a partial response. So 5 of our patients at the time of this data cutoff had a partial response, and their little circles went to squares at that time. So for this patient, it took a long time to get there. This patient, pretty quick, and so on. Other things to just note here, we tried a few different dose levels here. Patients had tumor shrinkage at all dose levels, regardless of what dose level they were. We had a mix of patients with beta-catenin alterations and APC mutations. It wasn't dependent on that. And then you're probably wondering if any of these patients were treated with gamma secretase inhibitors. They were. That's what these little hashtags are on the right. So even if they had prior exposure to that, they also had tumor shrinkage. So to us, this is like really impressive data. Like, we're really excited to see this, and we obviously are eager to treat more patients. Now, I do want to show some images. These are MRIs, so nothing too graphic. But I know some of my patients don't like to see pictures of things because it can be a little triggering. So if you're not somebody who wants to see pictures of desmoid tumors by MRI, then just maybe divert your eyes for a few moments here. All right, so these 5 pictures are 5 of these patients treated here. And these are all MRI scans, as I mentioned. And to orient you, they're all different parts of the body. Each patient's laying down and their head's going in their screen and their feet are coming towards us. And you can think of it like a slice of bread that we're looking at. So in this patient, this is somebody's neck, and this is their windpipe, this is their spine, and this white area here is the growing desmoid tumor. This is that first patient I mentioned. And this is their baseline scan before they got treated. I'll show you their treatment scan in a second. This patient has their tumor in their arm. This is their funny bone. So just a cross section of that. And then the tumor's right here. This one's the chest wall. So you may recognize this is the heart. This is the right lung. That's the left lung. And it's reversed because we're looking from the bottom. And then this is the tumor here along the chest wall. This is a tumor in the leg. This is the femur right there, cross section. And then this one is a large tumor that's in the abdominal wall, just below the belly button, kind of in front of the bladder, and a large tumor there. All right, so what happened to these patients when they went on trial and their tumor shrank? And you can see on this patient very clearly that it shrank there. Took a long time, but we got there. And this one shrank to that. This one shrank to this. This one. So I mentioned earlier that RECIST is a poor way to measure tumors. Like this tumor is like nearly gone, but by RECIST, they measure the longest diameter. It only shrank quote unquote 20%. But you can see like the patient had a pretty remarkable response. This one shrank. And then this one pretty impressively also shrank. So really, really nice to see. I bet a lot of you are asking, did patients feel better? And the answer is yes. Not every patient had symptoms, but pretty quickly when we put patients on the study, we can see improvement in range of motion, improvement in pain scales. We didn't formally measure that at this part of the study, but I can just tell you that anecdotally from talking to my patients. In later parts of the study, we are trying to capture that, so we'll have more concrete data on that as well. All right, I want to talk about side effects, but I'm going to take another little detour and talk about something called FAP, or familial adenomatous polyposis. So as I mentioned earlier, APC is an important driver of the mutations in APC is an important driver for the Wnt beta-catenin pathway, because that's the brake pedal that brakes on the system. So it turns out there's this syndrome where people can have a mutation that they're born with. It's inherited, where they have loss of function of that APC mutation. And patients with this syndrome can develop cancers because that beta-catenin pathway is, like I said, very important for that. And these could be real high-grade cancers like GI cancers, like colon cancer, and they can also get desmoid tumor. For any of you who are older like me, as you approach your 50s, you need to get a screening colonoscopy. And you're like, why would I need to do that? Well, the reason is to detect polyps in the colon, in the bowels, that could be precancerous lesions that could turn into cancer. So while an individual may get a colonoscopy and may have 0 or 1 or 2 polyps, patients with FAP can have hundreds or even thousands of polyps. And it starts when they're very young. So to prevent cancer from forming in these patients, they will often undergo removal of the colon, because that's where a lot of these cancers originate, and then they get kind of reconnected from their small bowel to their rectum. But even with that, they still have to be screened by an upper endoscopy, where they take a camera, look through the mouth to make sure they don't develop cancers in the small intestine. So in this cartoon here, this is the esophagus. So as you swallow food, it goes into the stomach and it goes into the small bowel. And this is called the duodenum. So patients with this inherited gene mutation will have to undergo screening endoscopies to look at the duodenum. Now, one of our patients had this procedure done before the trial, and there were a lot of polyps seen, and then they had a procedure done on the trial. So now I'm going to show something that's a little graphic. These are endoscopy pictures. So again, if anybody's medically queasy, just divert your eyes for a sec. But I think it's a really interesting picture and kind of proves that we're hitting this pathway, and I think pretty fascinating. All right, so 3, 2, 1, we're going to go. All right, so this is the patient's endoscopy before the study, and all these little arrows and circles are pointing to dozens of polyps that the patient has. There's a big one right here. And then on the study, they had a routine screening. This is part of the routine care. It has nothing to do with the trial. all these polyps are gone. And to me, this is pretty breathtaking to see this because we might be, we don't know, it's very early, but we might be changing the course for this patient's history as far as their risk of developing polyps or potentially a cancer. It's very early, it's we don't know that having these polyps goes away means that the cancer reduction's gone, but it's certainly very exciting and something that I would argue we need to investigate further. So, okay, so enough of that. All right, back to the study. So, what are the side effects? So this is a really common question, and I'm sure a lot of you are wondering this yourselves. We very carefully, as part of these studies, ask questions, 1,000 questions every visit about, what side effects they've had. We're checking lab values to see if they're abnormal. If there is a side effect that we already know about, we're asking if it's changed, and we're really trying to develop this. And we have a very strict way of kind of grading these and monitoring them over time. And if they're serious, it could change how we, conduct the study. So it's extremely important for us. So we are building that. The data I'm going to show you are from those 10 patients at this data cutoff. Now, if you're on the study and you're watching this and you're like, my side effect's not listed there. It's not that we don't know about it. We know about it. It hasn't been captured yet for public consumption. Okay. So the main points here are that like any drug we give, there's always side effects. It's pretty rare that we have a drug with no side effects. So most patients have side effects, but most of these were not severe, meaning they were kind of what we call low-grade side effects. So common things we saw: fatigue, nausea, change in some blood tests. We see some elevation in some liver function tests. We see some changes in sodium levels, interestingly. And then at the high levels, we do see hair loss. At the lower dose levels, hair grows back and there is not hair loss. At the doses that we're going to give patients in the future, and we're giving them now with desmoid tumors, there are no serious side effects. We have not had a single patient stop the drug for side effects for the data I showed. And importantly, for any trial, there's been no deaths from the treatment. For those of you who know about gamma secretase inhibitors, we run into some issues with ovarian function, diarrhea, rash. We really haven't seen any of that on this study yet. We do see some low-grade rash, but nothing severe. So what does this mean for you, and what does this mean for the trial? So as investigators, we feel like this has a very favorable safety profile and warrants continued monitoring, of course, but we think we can move forward with the trial and as we are. And as we build more data, we're going to get a better understanding of the more common and more rare side effects. If you ever participate in this trial, we're going to go through all those before you start, and you'll have a better sense if it's right for you. Okay. All right. So the last thing I want to do is just take you through that roadmap, and then I'm going to take all your questions. So when I see a new patient with desmoid tumor, the first thing I want to do is confirm the pathology. There's really excellent community pathologists. They probably have the diagnosis right. Every now and then we see something funny that might change the diagnosis. So I always like to have one of my experts take a look. If a patient's being treated in the community, that's totally fine. Community oncologists are amazing. It's incredible what they do. They treat so many different kinds of tumors. I do recommend at least once, and maybe ongoing, consulting with an expert. And for my patients who live far away, we can kind of work in tandem together. And then I'll make some kind of assessment if genetic testing is needed. I really don't want to miss that APC mutation. And if there's a strong family history of colon cancer, if somebody's very young, I often want to do that testing. And then the first question is treatment indicated? And this is always a hard one. And, a lot of desmoid patients, they have this kind of slow growing tumor. It's not causing any symptoms. It's not in a worrisome place. You can often watch people. And I'd say for most of the patients I see as consults, I'm just watching them. I'm not starting them on treatment. But if something's growing, if it's in a bad spot, if it's causing symptoms, then we got to make a decision on treatment. And we have a lot of options at hand. And so what I like to do is something called a shared decision, where we will work with your preferences, what we have available, sort of unique features about your tumor, and we'll pick something and we'll go for it. If we have a trial available, we're going to talk about it. If you've got a small tumor that we think we can do ablation on, we might consider that. That's where we, like I said before, freeze or burn it. Rarely we'll do surgery. We used to do surgery all the time, like when I started my career, but there's a very high recurrence rate, so that's kind of moved down the line now that we have better therapies. But there are rare cases where we'll still do it. And then radiation, we often don't do because there's some long-term risks of radiation exposure, like second cancers. But if I have an older individual very symptomatic tumor, we've failed other therapies, or maybe they've got other medical problems, and they can't get them, then we might consider radiation. And then, every step of the way, we'll kind of think things over. I think of it as blocks in time. We're going to do this for this amount of time. We might take a break. We'll do this till we get it under control. We might take a break. And you kind of ask this question as you go, and this kind of builds your individual roadmap where, you may start with observation. It may start growing. We put you on nirogacestat. That works for a while. You take a break for side effects. We may try sorafenib. We do that. It gets under control, starts growing. We might do a trial, and so on. And your story will be unique, but the hope is that we'll get control of this, stay ahead of it, and your quality of life will be high. And then hopefully at some point your tumor will just burn out, and that does happen. So when I stop sharing here, I know I talked for I'm a little over time, but I want to make sure we have time for
Yeah,
a lot of questions.
that was so great, Greg. Thank you so much. Just such important information for our community, and you just do such a great job presenting this. We do have a number of questions. We'll try to get to as many as we can. So the first one, and I'm just gonna, this is a good example of one that we have to remind everyone that there are some things that Greg can respond to as a clinician and other things that he's not able to either because the results of the study aren't in yet, or it needs to be an answer that are the trial sponsors responding to. So to the extent that you are able to, Greg, can you
Yeah.
comment on what do we need to know about zolucatitides or FOG-001's impact on bones?
Yeah. So beta-catenin is an important pathway for those fibroblast cells and those GI cells, but it's important for other parts of the body as well. And if we block that pathway, the way this drug works, we're going to hit some normal cells also. There's really no way to avoid that. So it turns out that the Wnt beta-catenin pathway is really important for bone health. And none of these trials, none of these patients on this data set that I showed you had any bone problems, but that's something that really could happen in theory. So we monitor patients with bone scans to make sure their bones are healthy, but we're also giving a medication to improve bone strength and to reduce that risk.
So does this mean that patients with osteoporosis are excluded from the study?
Yes,
Right
unfortunately,
now
yes.
they
Yeah,
are. Yeah, for their
that would
own
be. I think that would
protection.
be unsafe, to be honest. Yeah.
Yeah, and let's just take that for a moment and go. So, as a clinician you're as the medical oncologist and you're working with a whole team. Walk us through just a moment or two, like what decision do you go on from then? This person has osteoporosis. They cannot be on this trial. It could be something else that's happening. They can't be on this trial. How do you make a decision then about how this person will be treated if their tumor needs to be treated?
Yeah, so we'll go through the list of options and their
Mhm.
medical problems, and we'll try to make an assessment about, what's the easiest route to go. And often that's going to be gamma secretase inhibitors. I mean, the efficacy
Mhm.
of those drugs in those trials were great. We do have to watch out for things like rash and diarrhea, which can be severe in some people, and so we might, try that. And I find personally, I know everyone has different experiences. I find I'm starting drugs, stopping drugs, starting drugs, stopping
Yeah.
drugs, starting drug, reducing the dose, and eventually you can kind of find the right place for that patient to tolerate it. But other things work, and so like there's chemotherapy sounds scary. It is scary. I mean, these are powerful drugs, but a lot of the chemos we use for this are actually pretty easy chemotherapy drugs. Like we give them to children,
Mhm.
and so like I've had some patients who might be a little more frail that might tolerate it, liposomal doxorubicin better than they would a gamma secretase inhibitor. So we think about
Mhm.
that. And then for an older individual, like I said, like we will think about radiation. Like I've done that many
Okay.
times, if we need to.
Okay, that's great. That was one of the questions, if
Yeah,
radiation is still considered a
yeah,
treatment option.
yeah.
Okay.
But not somebody who's young. Like if you're in
Yeah,
your
right.
20s, like I'm not giving you radiation unless my back is against the wall.
Okay, good. And I mean, that leads to another question. Are children included in the FOG-001 or the
Yeah,
Zolucatetide trial?
yeah. Okay, so great. It's a really great question. So for a lot of phase 1 studies, we include 18 and plus.
Okay.
Sometimes for some drugs, we will, if there's a pediatric indication like osteosarcoma, bone sarcoma, we may push that down. Sometimes they'll say like skeletally mature. There's a lot of nuance in that, as you might imagine. But for this drug, given that we know beta-catenin can affect some of these other important pathways, we're starting at 18 plus for now. Um,
Okay.
as this drug gets more experience, I'm sure we will investigate this in younger populations. But for this point right now, we're not gonna we can't go below 18.
Okay, and again, this goes back to decision making. How do you think about decision making specifically between a gamma secretase inhibitor like nirogacestat or varegacestat, as we mentioned, which is in the pipeline for approval right now, versus zolucatetide, so or FOG-001?
Yeah, so that's a hard one. So when this started, right,
Yeah,
when we had only had a few handful of patients treated, the patients that were considering this trial were really people who've gone through everything. So
right,
that first individual I showed you, they had been on everything. There wasn't a single drug
right.
they hadn't been on, and nirogacestat wasn't FDA approved yet. So our back was up against the wall. We'd already just recently done chemo, and we decided to go for it. So
Uh-huh.
that was a pretty easy calculation. Now that we've built some data here and we see that this works, it's a little harder. So
Okay.
we have and it really comes up to the patient's preference and what they would be up for. So you have, a drug that, a lot of people have been treated with gamma secretase inhibitor that has a known side effect profile. It works. It takes some time to work. And you have this newer drug where we're seeing maybe some earlier responses that kind of hits this pathway right away, but a lot of unknowns, right? And a lot
Yeah,
into your schedule. So this is where the patient preference comes down. So like
yeah,
I've had some patients where they're interested, but the schedule was too busy. There's like no way I can
right.
do this. I've had some other patients who said, I want to see how the data matures. And then I've been very surprised. I've had a number of patients who said, I want to go right for this trial right from the beginning.
Right.
And that's an option too.
Let's just touch on that because there's a couple questions about like how do I even start this conversation or how do I even know what trials are out there? How do you
Yeah.
respond to that for someone
Okay.
who might not have you as their clinician, for
Yeah,
example?
yeah, great question. So, okay, so there's two routes. So, well, let's say three routes. So, one is, talk with your oncologist to see, what they know about for trials. And, as trials open, it's a pretty small community in the desmoid and sarcoma space. So we generally have a good idea what's open and what's looking good. And, if I don't have a trial and I know a friend of mine down in New York has it or something, I'm going to, refer the patient down there. Like we do this
Hmm.
all the time. Um, so that's one way to do it. There is a website called clinicaltrials.gov. I actually had a slide on this. I took it out for time space, but
That's okay.
yeah, clinicaltrials.gov. You can go there and you can put in desmoid, or if your
Yep.
family member has bladder cancer, you can put in bladder cancer, and you can click to see what trials are open. Now, I really recommend clicking off recruiting, like, to make sure that
Hmm.
you're filtering for that, because you otherwise you'll get a lot of trials that are closed.
Yep.
And then you can look around and most oncologists, if you bring a list, they can look at these and kind of tell you, oh, this looks good, this may not be good, or I don't know about this. And there's also contact information there. So if there's, a nearby medical center that has this trial, you can, reach out to that contact person and look at that. And the other spot is your website, right? Because you guys,
Yep.
that's what Tumor Research Foundation has listed all the trials.
Yeah, you can go directly to dtrf.org and it
Yeah.
takes you to clinicaltrials.gov. We do that, and then you do what Greg just said, right? In desmoid tumors, filter by location, filter
Yeah.
by what's active. But we do that because these are changing all the time. So in this case, with zolucatetide slash FOG-001, it's the same medicine for those who may have been in and out of the webinar. They're adding new locations, and so it's important to continue to stay abreast of that and to have that conversation with your care
And
team.
don't be shy. Like we
Right.
really want people to participate in trials because like we
Yeah.
we can't advance the field without that. So like if you're kind of on the fence, like just reach out and we can you can see see somebody who's involved and have a conversation about it.
Yeah, and I love what Greg you said, or Dr. Cote, you said, this is about
Very
shared
fun.
decision making. If we don't start the conversation with our clinical teams, then we don't get to that part about shared decision making. So show your clinical teams that you want that. I'm going to go back to the biology for a moment. I'm going to actually read this question. It's really important. You answered it to some respect when you talked about the genetic testing part of the decision making, but I just want to make sure this is addressed. If colon cancer and liver cancer are along the same lines in terms of the beta-catenin pathway, are they a hereditary factor in desmoids? There's, they go on a little bit. If we're looking at this pathway and there are hereditary factors, would this drug, speaking of zolucatetide, be most likely more effective than others?
Yeah. So okay. Yeah, that's complicated. So most colon cancer is not like uh, the, I'm not a colon cancer expert, but like there's lots of
That's
different
good.
ways to get colon cancer, right?
Yeah.
It's not just genetics. And we think that there's probably for cancer in general, it's probably a mix of your own genetics, exposures, and then probably just a bit of bad luck. Because
Yeah.
our DNA is constantly being bombarded with all sorts of insults, be it chemicals, radiation like UV radiation, other exposures, infections, and all these things can cause problems in that DNA. And you actually have like an army of proteins in every cell in your body to try to detect these mutations and try
Right.
to fix them. And so if they fix it, then the cell survives. If they can't fix it, they try to kill that cell. And if that can't work, then a cancer can form. So for people who have are born with problems that kind of predispose them to cancers, these are mutations in some of those genes, those proteins that are
Mhm.
important for protecting you against cancer. And so for the Wnt beta-catenin pathway for APC, that's just one of many, many, many different ways that your body tries to shut off this pathway and others to try to protect you from cancer. So it's a long way for me to saying that for most colon cancer, it's not because of an APC mutation that people are born with. It's just that pathway is active in that
Okay.
cancer. That's why it's an important target. And same with hepatocellular cancer. Like that liver cancer most commonly is driven by like toxic insults to the liver, hepatitis, alcohol, things like that. And it just happens to be that in the course of those forming, they can develop mutations in this pathway.
Okay. Okay. That's helpful. Thank you. And
Yeah.
again, all part of the conversation, the hereditary
Yeah.
aspect, the genetic aspect, all part of the
Yeah, and
conversation
I
with your
will say,
caregiver.
we're it's getting easier and easier to do genetic testing for the genes that people are born with, and at my institution, there's very strong advocates that most patients with cancer should have genetic testing for the genes they were born with, but it's a complicated conversation and one you should definitely have, with your oncologist.
This is an interesting question. I'm interested to hear your response, but we'll have to keep it really short so we stay on time. Do we know, are localized treatments such as chemoembolization and cryotherapy more successful than
Yeah.
systemic chemotherapy, or I'm going to throw in, or targeted therapy medications?
Yeah, great. It's a great question. So I'll say I'll hold off on the chemoembolization part because that's still a research question. But for the thermal ablation, so cold or hot, these are really useful tools. And the reason is because you can avoid treating the whole body when you're trying to treat just this, one tumor in this one area. And the issue though is that there's a lot of technical aspects of ablation that can make it successful or a failure. So one might be the size of the tumor. So if it's a really large desmoid tumor, it's hard to ablate that entire area. So the radiologists that I work with who do these procedures, they're pretty conservative. Like
Right.
they'll pick tumors that are either smaller or maybe the goal is just to alleviate some pain and not necessarily treat the whole tumor. There are some out there that are more aggressive and will treat a larger area.
Right.
So I think it depends on who you see. The other part is if the tumor is near a blood vessel that acts like a heat sink where it kind of draws away the cold or the heat, so it might be unsuccessful. And the other part is if the tumor is near a critical structure like the skin or nerve,
Right.
that ablation could damage those, and we don't want to cause harm either. So it really depends on the anatomy of your tumor. So if I see a patient with a small tumor and ablation is feasible, I'm definitely having that conversation because
Okay.
I don't want to subject people to medications that they don't necessarily need.
Right. And again, it's knowing the landscape of the treatment options and then understanding what's going to work for
Yeah.
you. As
And it,
yeah.
and it could also be part of a bigger treatment, like say,
Mhm.
say you start with this, but we realize we're going to have to do some systemic therapy
Right.
later. We do that and we want to try it again later. Yeah.
Yeah, I think that's a really important point for people to understand that these treatments may happen in combination or in succession with one another. Three more questions. I'm going to see if I can bring us home here. These come back a little bit more general. For patients who live in areas without local sarcoma or desmoid specialists, you touched on this a little bit, but whether in rural communities or outside the continental US, what options exist for connecting virtually with expert clinicians, and how do you recommend patients access that kind of specialized care?
Yeah, this is a tough one. So the U.S. law is a little convoluted here, where the for virtual visits the encounter there is happening where the patient's feet are. So if you're in Montana, like
Okay,
I would have to have a medical license in Montana
got it.
to treat
Okay,
you there, which is seems kind of crazy. Before the pandemic, I think that was a known rule, but not really enforced because there were so few virtual visits. But now virtual visits are very common, so it's harder and harder to do this. So I think the route is one of two things. Either you have a nearby oncologist who has a license out of state. So for me, I'm in New England. I have a license in other states in New England, so I can
Okay.
do that. Or we are allowed to have physician to physician consultations. So if you've got a really good community oncologist who's willing to work with somebody, they'll probably have a relationship with somebody who's seen desmoid tumors and other sarcomas before. And so they can call one of us up and say, hey, I got the situation. Is this okay? And that is allowed. So we often will do that. The other way to do it is just to travel like 1 or 2 times a year, or to do something like an e-consult, which is done on paper typically. These are
Mhm.
options as well.
Thanks, Dr. Cote. That's great. I'm going to read this one. It's really important. I'm interested in hearing and want to make sure that folks know that mental health is a very important part of this conversation. When a patient is initially diagnosed, do you offer mental health and social services support resources in tandem with medical and clinical information? We know that research has shown that the mental health toll of our diagnosis of having a desmoid tumor is
Mm.
significant and can often outweigh the the physiological impacts.
Yeah, this is a really, really important point. And desmoid tumors and cancer in general can take a huge toll, as you mentioned, on your psychosocial well-being. So we work very closely with a clinical social worker who's part of our sarcoma team, and not every patient needs her services, but those that do, like she's available for consultation. We have psychiatrists who specialize in oncology who are also available. And so we work together with that group to try to support people as best we can.
Thanks, Greg. And a plug for folks, patients and caregivers talking to their oncologist and their
Yeah.
medical team about referrals for mental health, for nutrition, for physical therapy, for whatever is needed. As a desmoid tumor patient, you have a right to be opening those conversations. Please do so. Um, Last question. We didn't get to every single question, so please a reminder that info@DTRF is available to you, as are all of the resources on our website, dtrf.org. So, Greg, bring us home with when you look at where desmoid research and treatment are headed. Do you feel hopeful that we may one day have curative options, and what advances seem most promising to you right now?
Yeah, I am so incredibly hopeful right now, and
Mm,
I think in general for the oncology space altogether, because a lot of things that I thought were undruggable, like we're seeing people figure out how to do this, and Zolucatetide is really just the first step in like these small molecule
yeah.
proteins that can get into spaces that we didn't think we could get a small molecule before. So I'm really hopeful that this could be the start of something really big in not just with this company. There's many other companies that are working on this and on these kinds of ideas. And so I am hopeful we'll get there. It's going to take some time. Like like everything, things are slow growing, but the science is really really progressing quickly. So I think we'll get there.
That's so great, Greg. And I think, this is really one of my key takeaways. And I feel this in almost every conversation I have about desmoid tumors with clinicians, with clinician researchers like you all, like you, with scientists, is that a collaboration is key. And in the desmoid tumor space and in the rare disease space, collaboration is even more key. And we have an incredible group of clinicians and researchers in the desmoid space that care about all of you, each one of you that is on this call, and are really working tirelessly to find the answers needed to address this disease. And so that's a big takeaway for me. Another big takeaway, and it comes to a question that just popped up. Do I have to bring up clinical trials as an option to my care team? They didn't bring it up to me. So the other takeaway that we've heard from Dr. Cote in various different ways is yes, please bring up conversations to your care team. You have a right as a patient to do that. Bring them your what you see on clinicaltrials.gov. Use the resources on DTRF. Start the conversations. You have a right to do that. We are just about at time, so I just want to say thank you so much for these valuable questions from our community and for the incredible presentation and responses we heard from Dr. Gregory Cote today. I also want to mention that these webinars are made possible by your generosity. We are a nonprofit at DTRF at the Desmoid Tumor Research Foundation, and so it's your gifts that support educational programming as well as our funded research grant program. If you are in the space to make a gift today, any gift matters. Every gift makes a difference, and you can do that at dtrf.org. Before we get off, I want to let you know of a couple upcoming events with DTRF. Number one, you are invited to join us at our signature event of the year, which is the Together We Will weekend featured in my background here, taking place on September 25th to the 27th in Philadelphia. Registration is open at dtrf.org. Also, please keep your eyes open for a registration for our next webinar, which will continue to talk about treatment options, decision making, and we'll focus on the drug option varegacestat, which is another GSI that is important because it is in the pipeline now to be another approved drug. That will take place on July 14th. And registration will open in the upcoming weeks. You can find more information about these events as well as a lot of information on our website, dtrf.org. So I'd like to extend a huge gratitude to our presenter, Dr. Cote, for being with us today. Dr. Cote, Greg, thank you. Thank you. Thank you for your dedication to the desmoid community, and please keep going. Our mission, as you know, at DTRF is to find a cure for each and every desmoid patient, and it's clinicians like you that are moving us closer to that every day. And once again, I want to say a huge thank you to the community for being with us today and for being attentive to all of DTRF's work and offerings. Please keep talking about and learning about our rare disease. Continue sharing accurate resources, engaging with DTRF content, and helping us raise awareness wherever conversations about desmoid tumors are happening. The recording of this webinar will be available shortly and posted on our video library. And thank you once again for being part of the DTRF. We're wishing you all a peaceful evening. Take care.